Hello, everyone, and welcome to the second part of lecture number 2 here, continuing along in chapter 26 lecture series here. We're continuing through the nephron here, and in this case, we've just finished up the renal corpuscle, talked about the process of filtration, and talked about the regulation at the renal corpuscle. But now we're going to move to the second part, and as you can see as we get down here, the second part is going to be the proximal convoluted tubule, abbreviated PCT. And then from the proximal convoluted tubule, we go down the nephron loop here. And so that's what we're going to be talking about. We'll save the distal convoluted tubule and then, of course, the collecting duct for a little bit later. But this is what we're going to be focusing on is the next two segments. And as we saw, the first segment, taking approximately 30 minutes to lecture over, quite involved, but we'll see some of the similar stuff. I'm going to stress the main points. We'll talk a little bit about countercurrent multiplication, which occurs at the nephron loop, but it's a concept that students are expected to know, but again, I don't test too heavily on it. It's really a complicated concept, so I tend to keep the test questions kind of basic there. All right, so let's continue along this. Again, make sure you have this anatomy down. That's really what was lectured on the first part of lecture number 2. You really need to know this, but we're going to move ahead to here. And hopefully you're seeing in these figures here, we're dealing with simple cuboidal epithelium, and it does have some microvilli. That's what all these little lines are. But let's get into this. As usual, if you want to read up on the objectives, these were the objectives that we've covered quite a bit. And then as you can see down here, we're just focusing on PCT and the nephron loop and a couple of main concepts to get through there. So let's go ahead and get into this lecture here. So we're finally starting to get into the nephron tubules here. And the nephron tubules is going to consist of the proximal convoluted tubule, right? And the distal convoluted tubule. So that's where the term tubules technically comes from. I tend to group, when we talk about the tubes, I tend to include everything else, including the nephron loop. It's some minor classification differences. Some people might argue that the nephron loop is the loop and not part of the tubules. I'm not that specific on my content, but just be aware some people might consider the tubules to only be the PCT and the DCT, and the nephron loop might not get categorized there. I don't stress the differences between them, so that's kind of what we're not really going to get too focused on. But anyway, let's really get into the main point. So what's after the glomerular capsule, often abbreviated GC, or what's after the renal corpuscle? Because again, renal corpuscle is this whole thing. The glomerular capsule is this little space here that we're talking about. Okay, and so what's after it is the PCT, or the proximal convoluted tubule. That's the most adjacent to either the glomerular capsule or most adjacent to the renal corpuscle, however we want to see it. Again, renal corpuscle, don't forget, is the capsule and the glomerulus as a whole. Otherwise, if we just said the glomerular capsule, we're talking about this inner space here, where certainly urine's going to form. So a couple of minor differences between those terms, but nothing major. Certainly, if I use glomerular capsule and look for that as an answer, I wouldn't include renal corpuscle. If I was looking for renal corpuscle as an answer, you wouldn't have glomerular capsule. So that's kind of the trade-off that we have there. All right, but hands down, this is the main point that happens at the proximal convoluted tubule. Most reabsorption, since it's 60 to 70% of the reabsorption that occurs at the PCT, this is what the main point is most reabsorption occurs at the PCT. So if I ask any questions, or any instructor asks questions about what's the main event at the PCT, it's the site of most reabsorption. So filtration happened at the renal corpuscle. It's the only location where we have filtration. So when it comes to reabsorption, most of it's going to happen at the PCT. Now, that doesn't mean 0 happens along the loop or 0 happens along the DCT. It's just saying most of it happens at the PCT. That's the main point. This right here is what you should note. Okay, so the PCT tends to be an area that I test quite a bit on. Now, to understand what's happening here is this bullet point here. So you don't need to get caught up on this. This isn't something I'm going to describe in details and ask a question about. But I think it's important to understand what's going on. So here's the filtrate. The filtrate coming from the renal corpuscle would start to enter into the tubules here. So now we're in the PCT here, and we know that because we're dealing with simple cuboidal epithelium with microvilli. So notice the cells of the PCT, the type of epithelial tissue I expect you to know is simple cuboidal. Okay. Now what happens is the filtrate, substances will pass through the filtrate over here. That would be this region here, and you can see these are called co-transporters. Notice when sodium moves in, amino acids move in. When sodium moves in, glucose moves in. So notice sodium is a key co-transporter. So it transports it. That's the first step. Fluid leaves tubular fluid, and enters into the cells of the PCT. Here's the tubular fluid. Here's it entering into the cells. From entering into the cells, we'd be in this middle spot. What's going to happen is the fluids, or the solutes and the fluids, will leave and they'll enter into, notice, what's called paratubular fluid. So that's right here, also known as interstitial fluid. So notice paratubular fluid, peritubular, between the fluids. It's not tubular fluid, it's paratubular, that's between. Or you can note it as interstitial fluid. So that's all we're saying. This fluid outside of a tissue and outside the blood vessel tends to be interstitial fluid. Now that's eventually going to diffuse into the paratubular capillaries. That's what we're seeing here. So this is just showing you when we talk about reabsorption, what's happening is reabsorption is removing the filtrate from the actual tubes. Okay, so the filtrate enters into this, the actual cells. Then from the cells, it enters into interstitial fluid. Then from interstitial fluid, it enters into the blood. Because again, we're talking reabsorption. So we're getting things back into the body. That's what reabsorption is. Otherwise, if we were to do the opposite and we're going to focus on secretion, secretion would be removing it from the blood, entering into interstitial fluid, then entering into the cells, and then eventually entering to be secreted or removed from the body into the fluid or into the filtrate. Okay, so these are the concepts that we're dealing with. Again, I don't necessarily test on this. The cells of the PCT, I certainly is fair game. But as far as describing this flow, it's important to know, but it's not something, I would necessarily test on. All right, so the filtrate, once we get through there, because we had filtration, it's now considered tubular fluid. So notice filtrate flow is going this way. Eventually that filtrate, because we're now in the tubules, right? We're in the proximal convoluted tubule, technically called tubular fluid. Usually I'm not too big or too specific on these fluid names. I will use them appropriately on the exam. So it is important that students recognize the terms and what it implies. But I wouldn't get caught up on a direct question like what is tubular fluid or where does tubular fluid start. It technically starts in the proximal convoluted tubule. All right, but a key point, if you notice, we've been showing you sodium gets reabsorbed. So sodium ion reabsorption is something that's really important. Notice you need to know what's the role of sodium. Sodium is important for co and counter transport mechanisms. So here we're seeing the co-transport into the actual fluids there. But here what we're seeing, this is counter transport. Notice active transport, that's the sodium and potassium. Here we see a symport, it's potassium out and chloride out. Here we see facilitated diffusion. These are just individual things using. And here we can see osmosis. And most of the time water is moving by osmosis. But if we look down here, it's all symport or co-transport. All right, so co-transport, you should know, is the movement of two ingredients in the same direction. Counter-transport would be moving ingredients, one ingredient in and one ingredient out. Now, understand this role, that's the importance of sodium. Sodium helps with a lot of the co and counter-transport. I expect students to know this. So far, really from testing material, it's knowing the main site of reabsorption is the PCT, knowing that it consists of simple cuboidal epithelium, and then so far knowing that sodium is really important for co and counter transport mechanisms. And so we're going to get into the 5 main functions of the PCT that you should be aware of. Now, before we get into those 5 functions, just keep in mind that because these carriers are so important, that if you have an inefficient or a defective glucose carrier in this region, understand that that means our body can't reabsorb the glucose. And if we don't reabsorb the glucose, that means that glucose stays inside the filtrate and inside the tubular fluid and eventually becomes urine. And that would be glycosuria. So know that that's high glucose in the urine. We've seen this term before. We've seen glucoseuria. Now we're seeing the term glycosuria. It's basically the similar sort of effect here, just knowing that it's high glucose in the urine. So you should be able to recognize it. In the case, since we're talking, of course, the urinary system, understand that its relation to the urinary system would suggest that there's a problem with glucose transport at the PCT because, again, the PCT is the main site of reabsorption. All right, but let's start breaking down the functions of the PCT. So this figure here, I believe, is one of the unique figures that I took from McGraw-Hill. So they had a little bit better of a figure showing some of these. The book image here, this is our Pearson's image. And as we can see, here's the co-transport. Here's the transport just by itself. Here is counter-transport. So this book does a really nice job, I think, showing all the different carriers. Here they do a nice job showing you some of the counter-transport, which they didn't show there. There's the for bringing in sodium, notice there was hydrogen exchange. So there is some pH balance. We're going to talk about pH balance a bit later in this unit. All right, so let's get into the function. So again, main function, reabsorption of organic nutrients. That is the main function. Again, site of most reabsorption, and the things that get reabsorbed here are 99% of our good ingredients. That is the main point. That's your main walkway. That's what you need to understand. Now, also note that this reabsorption of our main nutrients occurs via facilitated transport and co-transport. So here, this is facilitated. We can see there's some facilitated down here, but this is showing you just straight up diffusion, and then this is straight up diffusion. But if we had proteins down here, that's where we're having some of that facilitated transport. This is actually co-transport. The main point is you should know when we're talking about absorbing glucose amino acids, i.e., we're relying on facilitated transport and co-transport with sodium, which just continues to build off of this point here. Sodium is the main ingredient moving a lot of our important substances, as we see. Otherwise, notice facilitated diffusion. So you have to use both figures, and that's why I had both figures on here. I feel like both publishers did a good job with the figures, but kind of cut the figures a little bit short on what should have been shown. So if you combine both figures, making sense, here's your facilitated transport or sim transport. Here's your facilitated transport as well. Again, a lot of times it's with sodium. That's the main point. If you got this down, site of most reabsorption of all the good ingredients, and that it occurs by co and counter transport or facilitated transport in general, you're great. All right, the second main point is there is some regulation of pH. Now, we're going to come to this later. You should know that this occurs, and it's going to occur at pretty much every stop moving forward, or I should actually say at the DCT and the PCT. It's not going to occur here, but along the PCT and the DCT, we do have regulation of pH. And so what you need to really understand, and the concept that you need to work to, is understanding that when we have, when our pH is acidic or our blood is acidic, we're going to secrete H+, meaning we're going to get rid of H+. And then when our body is, when it's basic, so if we had an alkalines, we would be getting rid of this bicarbonate. So notice when we have a low pH or an acidic pH, we get rid of hydrogen and we absorb the bicarbonate ion. This ion will help us reduce, will actually help us increase the pH. So again, it will increase pH having more of this. And if we needed the opposite, so if we were alkaline, we would reabsorb hydrogen and we would secrete this bicarbonate. We're going to come back to this later. So for now, know, again, main site of reabsorption is your main point that you should be walking away. Now, step number 3 is a linked process. And this is the hard part. This is one of the higher detailed questions. This is the area that, this is really where the A student gets the point and the B student just kind of misses this one. So this is the highly detailed part. So understand there's linked absorption. So what has to happen first is sodium gets actively absorbed, okay? That active absorption of sodium is going to drive other ions and then drives osmosis, okay? So sodium gets reabsorbed, that drives the movement of other ions, and that also drives osmosis. So understand we absorb ions, then we absorb water. So notice 90% of water occurs via osmosis, and that's how water always moves in our body. It always moves via osmosis. All right. So the key ion that will drive ion reabsorption, what ion do you think is going to drive this process? Hopefully you got it down. That sodium is super important. So sodium functioning as a symporter drives this process. All right, what hormone regulates the sodium reabsorption, or what hormone increases sodium reabsorption? Hopefully you remember it was aldosterone, but really what we didn't talk about was the role of angiotensin 2. So what you need to know is angiotensin 2 targets the PCT. Okay, so angiotensin 2 targets the PCT. That's the main hormone that I expect you to note. And what that does is that's going to stimulate sodium reabsorption. Now that we get sodium reabsorption stimulated, sodium follows along and we get some co-transport. And then because we're absorbing ions, water follows behind it. All right. So we're continuing on with this. So we're talking the ion and water balance. So we're still on bullet point number 3 here, the absorption of ions in water. So main point is, first, we have the sodium-potassium pump that keeps sodium concentration low inside the tubular cells. So these cells, because we have a sodium-potassium pump right there, we're doing this, we're keeping sodium low, okay? Second, the symporters at the apical surface use the concentration gradient to drive solutes out of tubular fluid. So here, now we're driving out our our ions, okay? So low sodium here that allows ions to come in, then we push them into essentially the interstitial fluid there, right? And that's this part right there. Now that we've got a buildup of salts here, right? Because that's what we're doing. We're building up salts in this area. Now that drives osmosis. So sodium gets pumped. We keep sodium low in here. That allows for the buildup of salts. Salt concentrations out here are going to be lower, right? But sodium will be high here, but we're using an active transport, so that's fine. And so now we're driving the other solutes, and then eventually water follows. All right, so osmosis reduces the solute concentration here. And so now we have passive ion reabsorption. So now water dilutes this. Okay. And so that's kind of the exchange. So notice this is a really, really complex process. The main point of me getting into these details is to show you the complexity of this. Don't get caught up on it. It's not necessarily something that I get into a ton of details. Like I said, I expect students to know salt absorption happens, that drives ion absorption, and then that drives water absorption. If you get that down, that's the main point to occur. So notice I listed these main points. Number one, you should know site of most reabsorption is definitely the PCT. Number two, you should know sodium ion absorption is the driving reabsorption process. It drives it because it does co-transport and counter-transport. Number three, PCT is the site of angiotensin II, which helps with the sodium reabsorption. That's your third big point. And then finally, you should know that pH balance occurs here. And it occurs when we have a low pH, we're going to secrete hydrogen and reabsorb bicarbonate. Or when we have a high pH, we're going to reabsorb hydrogen ions and get rid of bicarbonate. Okay, again, the pH balance we'll come back to later. You should be getting those points, but if you wanted to know that again, because we are going to talk about it and you are going to need to know it at some point. But again, when we have an acidic pH, we secrete hydrogen and we reabsorb the bicarbonate. When we have a high pH, we reabsorb hydrogen and we secrete the bicarbonate. If you want that on your notes, add it now. Otherwise, just wait. We'll come back to it. So again, major points. If we go through this whole part and we look at our main functions. So this is it, main functions. Main site of reabsorption. We can regulate pH. You're going to need to learn that process later. You should note that this is the PCT is the site of angiotensin II, and that the way absorption occurs is we absorb sodium first. That drives the ion absorption of other ions. As ions build up in interstitial fluid, that's going to drive osmosis. Osmosis will dilute the ion concentration, and that'll result in passive ion flow. So it's active sodium reabsorption. So notice active sodium reabsorption because we're talking a pump. Symporters to drive the others. So here's the others getting driven. All right. And then ion gradient now drives osmosis. Osmosis reduces and helps passive ion concentration. So it's active sodium ion pumping, followed by symporters to move other ions, followed by osmosis following those ions, followed by passive movement of those ions. So it's a four-step process. It begins with active movement of sodium, symport movement with sodium and other ions, osmosis, followed by passive movement. Again, hard for me to test on your major points to focus on as far as four main questions to ask or discuss or four key concepts. It's really right in those main points. So really focus on that. Like I said, this is a hard chapter. It's a hard unit. It has a lot of hard questions. So keep focused on it, but the major point is reabsorption, angiotensin II, and the role of sodium. Focus on those things. Don't necessarily get caught up on this. This is really hard for me to create great questions that I feel is fair game for students. All right, but this brings us to the nephron loop, and I believe this is also another image from McGraw-Hill, not necessarily Pearson. Pearson has similar images, but notice this is the countercurrent. Notice what direction filtrate is going is the opposite direction of blood flow, so we're going to come back to that. So At the nephron loop, keep in mind 60 to 70% of our ions and those minerals have already been reabsorbed already. Okay. At the loop, we go from simple cuboidal to simple squamous. What's not shown on here, but if we look back here, notice there's a thick segment. So this would be the thick descending loop. This is the thick ascending loop. And then notice there's thin segments. So we're going to talk about those different segments at these loop. So where it's thick, it's going to be simple cuboidal. And then where it's thin, it's going to be simple squamous. So keep that in mind. All right, so at the nephron loop, we're going to continue to absorb water, and we're going to continue to absorb sodium. We're going to continue to absorb some potassium, and we're going to absorb some chloride ions along the way here. All right, and so I'm going to be really blunt and really specific with what you need to know here. So when we get to the thin descending limb, so notice it's the thin descending limb. Notice I didn't really talk about the thick descending limb, so don't worry about that one. We're going to focus on the thin descending limb. So notice the thin descending limb, you should know, is simple squamous epithelium, and it's permeable to water only. So solutes are not going to move. So we're talking right here, water movement is the main area. So notice water reabsorption occurs there. And if you're looking at this figure does a nice job explaining what's going on, talking about the vasa recta. So mostly here, if you notice, they're using the term vasa recta, which tells you they're mostly talking the juxtamedullary nephron being the main one. It doesn't have to be, but because the juxtamedullary nephron goes deeper into the nephron, into the medulla, there's going to be a reason. We're going to talk about that in a second. But if you notice, they're showing you we're having osmosis occur, and what you need to really know is water is reabsorbed. So for the thin descending limb, it's really, really simple. If I ask any question about the thin descending limb, it's 2 bullet points. It's simple squamous epithelium, and it's the site where water gets reabsorbed. So when water is getting reabsorbed here, what you need to understand is water is getting reabsorbed, and therefore the filtrate is now getting concentrated in solutes because we just got rid of water. When we start going, so this was the start. Here's the renal corpuscle. We move through the DCT. We're flowing down this way. Now notice, one thing to point out is the filtrate's going this way, but the blood is going this way. And the reason that's important, and we're going opposite directions. The filtrate's going one way, blood is going another. And the reason that's important is because we're going to remove fluids from this area. So as this is getting carried away, if we build up solutes here, it can get absorbed into the blood and take those solutes away. Again, another complex area. It's an area that I'm going to discuss in quite a bit of details, but it's not an area that I test super heavily on. And again, I say this because I need to get into the heavy details. You need to get some exposure to these kind of content, and you need to work through these details. It's that simple. Those details will help you understand the basics. So right now, thin descending limb, do you know water is reabsorbed there, and do you know it's simple squamous epithelium? That's really all you need to know for the nephron loop so far. All right, so then the next section we go into is we go into the thin and thick ascending limbs. So that's this part here. You can see the thin ascending limb is a small segment, and then it becomes thick ascending limb pretty quick on. All right, so what you need to know is the thin ascending loop is permeable to sodium, to potassium, and to chloride ions, and it's simple squamous because it's part of the thin. So anytime we use thin loop, all right, it's going to be simple squamous. All right, notice this is permeable, it's passive flow. So because it's simple squamous, we can have passive flow going through there. All right, and so here they're showing you the thin segment of the ascending limb of the loop is not permeable to water, but is permeable to solutes. So notice these are solutes being diffused here, where here we had water diffusing in. I don't really know why they showed too much solute diffusion here. There really shouldn't be any. I'm kind of surprised that they show it. So you can kind of scratch that off. But this solute transport is going to definitely happen. This is minor. I would ignore that. When I talk about the descending limb, it's always water reabsorption. That is it. So ignore the figure here. There's a chance that some can, but we're not going to focus on that at all. Then the ascending loop is where I want you thinking sodium, potassium, and chloride. Now, the thin loop is just passive flow. And you can see, again, they're mentioning the vasa recta. All right, but notice the solutes diffuse out of the tubule and into the more dilute interstitial fluid, right? Because that interstitial fluid, we just passed water into this area. So now that's allowing for us to build up solutes. And again, it's because of the countercurrent flow. Notice this is going up and this is going down. So the fluid is moving down this way as far as blood goes, while this is moving up. All right, so the thick ascending limb you need to know is active transport of sodium that helps establish the gradient, and that's what we use to pump sodium, potassium, and chloride. So notice the ascending limb is always sodium, potassium, and chloride. It's just the thin limb is passive, and the thick ascending limb is going to be active transport. Okay, so know that. And as we go up, as you can see, as we're going down, was water flows out here, it dilutes this area, and then as filtrate flows up and starts pushing out substances here, it reconcentrates the area, which is really unique, and that's why we keep stressing something called countercurrent multiplication. So that's what this is called, countercurrent multiplication, and what that really means is the effects are additive because of the fluids moving in opposite directions. All right, but I'm going to keep this simple. I'm going to try to keep it as simplistic as I can. And what that means is when we talk countercurrent multiplication, this is what its main point is. It's going to keep the solutes concentrated in the medulla, which helps concentrate the urine. If you remember, I was pointing out that they weren't calling these the peritubular capillaries. They were calling them the vasa recta. And if you remember, vasa recta is unique to juxtamedullary nephrons. Those are the loops that really go deep into the medulla. Well, the reason why we talked about that is it was a way for concentrating urine, right? Those juxtamedullary nephrons can form concentrated urine. It's because the loop is deep down in there. So this deep loop, you should understand, high salt concentrations are going to be found in the medulla. All right, and so what happens to understand this is, one, water flows out of the descending limb. We just talked about that, right? What that does is it dilutes the peritubular fluid. The peritubular fluid is in between, so this gets diluted, and what that does is it increases the concentrate of the filtrate. So here, we dilute this area, but at the same time, now our filtrate is full of solutes. Or what we're doing is we're diluting the filtrate, we're diluting the interstitial fluid here, but now notice the filtrate itself is actually full of solutes. So now once we round this area, well, now we're going to want to push solutes out because we just diluted it. And that's why it's really, really weird. So notice at the loop, less water is going to leave and solutes are going to enter. The tubular fluid at this point and the filtrate concentration will be equal. And that's really why we're not going to have much exchange going on here. But notice they're just showing you the osmolarity in between here. So we dilute it on the way up, but at the same time, we dilute this area on the way down where water is moving, but on our way up, we start to add solutes back, and so those solutes kind of form in this area. All right, so the concentration of filtrate is high as it ascends. So as it's going up, the solute concentration here is going to be high, and that's when sodium and chloride and potassium get pumped out. So I am missing potassium on that list. All right. The main point of countercurrent multiplication is really this. It results in a high concentration of salts in the medulla and low near the actual tubules, meaning the PCT and the DCT. As I mentioned earlier, some people consider the nephron loop as part of the tubules, but really we should be thinking PCT and DCT is the tubular network, and we should think of the loop as literally the loop. All right, so it's going to be low near the tubules, and that's what allows us to concentrate urea and waste within the tubules. So notice the function of the loop of Henle is to form concentrated urine within the loop, and that allows us to actually have by the time we reach the DCT, it allows us to have diluted it a little bit for the final exchange, right? Because we just pushed out actively transported sodium, chloride, and potassium. By doing that, we have now diluted the urine to allow the final site of exchange. So that's the main point of the ascending limb: sodium, potassium, and chloride. I know this is super complex. I know we're getting into a lot of details. Hopefully you're walking away with the big picture. The big picture is the descending limb is permeable to water. The ascending limb is permeable to sodium, chloride, and potassium, which we see right down here. To understand the whole process, you're going to need to read through all that. But fortunately, I don't test as heavily on this process as I probably should, or as other instructors might. I would read through this, really understand what's going on here. But again, water moves out here. This water dilutes this lower area that allows salts to start it being exchanged. And then as it comes up, notice this area is diluted. So as we move salts out, it's fine because we have a diluted area. But by the time all this filtrate as it moves upward gets diluted, well, now we're having equal concentrations. And so now we're going to have our final exchange at the DCT, or distal convoluted tubule. All right, so let's review the main points of the nephron loop because I know it's quite complex. But again, just focus on the descending and the ascending limb. That countercurrent multiplication is a big concept that the state requires us to test you on. But the reality is, just know that it helps concentrate salts in the medulla, and that helps dilute the actual filtrate on its way up. All right, so main points here: site of water and sodium potassium chloride reabsorption. The thin descending limb is where water reabsorption occurs. The thin ascending limb is passive sodium potassium and chloride reabsorption. The thick ascending limb is active sodium potassium and chloride reabsorption. And we're going to call this all active because we need to use energy to move sodium, and therefore chloride and potassium will not move without the active movement of sodium. So we're just going to call it all active transport. Area where countercurrent multiplication occurs. You should know it's the nephron loop is the site of countercurrent multiplication. And what that countercurrent is, blood is flowing in one direction, the filtrate is flowing in the opposite direction. So that's the countercurrent part. The multiplications part is it helps concentrate solutes in the medulla because, again, we dilute it and then we end up diluting it, but then we end up concentrating. We dilute it with the water, but we concentrate it with the active transport of sodium and potassium. So very interesting, very complex. Don't get too carried away. Like I said, the main point is countercurrent multiplication allows high salt concentration in the medulla and low salt near the tubules, which allows the tubules to perform their functions of reabsorption and secretion. All right. Overall, that's going to help really get our waste in there. Basically, this was all about salt and water balance. That's the nephron loop, salt and water balance. Okay, so focus on really this area here and this bullet point down at the bottom, and you should be fine with the nephron loop. Those of you that really want to understand the concept, I would read up on those figures, okay? I would read through this. It's going to take a lot of work to really understand it. It is a complex process. Like I said, the complex things like this, countercurrent multiplication, would probably be one single question over this, even though we spent much more time describing all these concepts. So don't get caught up on countercurrent multiplication. Just have a minor understanding of it. But hopefully you understand that. And if you notice, relate it to juxtamedullary nephrons, because those have the deep nephron loop and the vasa recta, which is exactly what they're describing down here when they're talking about the vasa recta there. Okay? So really pay attention to those things. They do, like I said, the figures do a pretty nice job describing it, but if you know these points here and you're able to summarize that, I would say you have 4 of the 5 questions down, or 3 of the 4 questions down, how many ever I'm going to ask, as long as you have those major points down. All right, but at this point, obviously, you should be able to answer all of these basic group note review questions. Really, this lecture obviously focused on the PCT and nephron loop, but part 2 of the lecture, of course, was all about the renal corpuscle. So we are done with lecture number 2. We finished both parts here. As usual, thank you all for listening. I hope this is making a bit of sense. This is one of the harder lectures. Lecture 2, and going through the nephron and understanding the main points of each part is really the challenge. So stick with it. We got a couple more parts to get through here. So we're moving on to lecture number 3. Thank you all for listening. I hope you have a great rest of the day. Keep at it as usual. If you have questions, please do not hesitate to reach out to me and get in contact with me some way. Take care, and I'll see you at the 3rd lecture coming up.